interesting questions for us who are from the side of it, using AI, at least as partially a tool in our daily work. How do you see, after this discussion about an expertise that you have, but on the bench side, do you see it as bringing value? Do you see it as a threat? Do you see it as a noise that is disturbing the way you can interact with data coming from the other world?
It's just changed the paradigm and the way we are designing drugs. So meaning that, even it was not the case anymore, but we don't screen anymore 1 million or 2 million of compounds, but we are more reasonable volume of compound. But I think we can learn and work. It's really a collaborative way of working because we can benefit from each. Even if we test less compound, we have to test them faster. We have to reduce even more the variability because computer and AI model doesn't accept the variability. And by nature, biology is variable, even if we do our maximum. We have to be even better to generate data and more data because consolidated data, a mean of IC50 value is not enough anymore. And we have to give more data, better quality, more robust, and faster because the cycle accelerates a lot. I really see now it as a partnership, and I will be always being skeptic, but positively skeptic on what the AI model will tell me about my data that I generate. But that's no, it's a win-win situation, and that can help also to if we think about cellular complex model, cell painting, or high content imaging. There is so many data generated in one image. You need the support of the AI to analyze your data anyway. So that's that's the new brother or sister in the family you have to learn to deal with and collaborate.
I think the right word is really collaborate, because I don't see this just as an old-fashioned of saying we have new technology and well, first of all, you are in the right place because your data never lies. Our data is made up, right? So it's always questionable. Your data is robust and there because it's experimental. So we have to respect that. But the added value is not really just to say, oh, we predict something, we think it's this, test it, but really work together. So the key word is really collaboration. And we should not resort to experiment as the moment where we should ask whether or not what we predict is correct, but build together, somehow put the know-how entirely in a working life cycle between exchanging data and working on it, instead of just resorting to a final approval of our prediction. I think that people who work that way are not in the right path because they lose time and they lose the opportunity to build on solid experience that is needed.
I think there still are plenty of instances where even in the chemistry realm, the synthetic data is not easy to generate. And so in cases, there are opportunities for active learning where, in fact, to test molecules primarily for the purpose of filling in holes for data modeling, for data that would probably have been a request that you never would have gotten 5 years ago, right? But now perhaps your work can be augmented with that. Also, with respect to more complex models that we believe will better reflect disease states and direct us toward the right molecules to modulate them, if you have the capability to generate cell-based models of various disease states because of decades of expertise in this area, it seems that is an area, right, for growth.
Yes, especially now with the high throughput and stuff. Coming back to the experiment for phenotypic screen, when we do the gene induction, then we add the exact amount of molecule having a label activity that is similar to what we test, but they are inactive in the test. So we use the gene induction data of the inactive compound to test the hypothesis that we do analyzing the pathway emerging this exercise, just because we want to test within the experiment the hypothesis of the mechanicistic that we found. That is the inactive one. So seems to have active on the same target as a label, but is inactive in the phenotypic assay. So the genetic fingerprint had to be different, otherwise it had to be active. So if we don't have this data set that is big enough to support the identification of the actual driver of the phenotypic screen, then we miss something. The hypothesis that we invest triple the experiment afterwards to validate. In this case, by design, we add information that we know that is not good in the direction of the activity, but is good in deriving or validated model by the beginning of the story. So this is a way that we have to change thinking. So the model, if it's driving, had to drive. Otherwise, is a part and could be a problem, it could be something that is misleading because it can be wrong. So had to enter in full cooperation with the experiment to be really embedded in your strategy and tactics. Otherwise, it's something like in the past that is good to have. My boss says at the very beginning of my career, you are the new guy is making the picture for my JMedChem publication. And the story changed a bit today, but I mean, the mindset and the work that you put in place is fundamental as much as the quality of the model of the experiment.
That's awesome. Well, thank you all very much.
Thank you.
Thank you.